Welcome to the second part of our lecture on the follicular phase of the estrous cycle. During the follicular phase of the estrous cycle, ovulation usually occurs. In ovulation, there is an increase in the LH. This is also known as the preovulatory LH surge, and this surge will trigger ovulation. So this is a diagram of the follicle, and the oocyte is located in the center. The oocyte is surrounded by the cumulus cells, also known as the cumulus oophorus. And for the oocyte to get out of the follicle during ovulation, it must cross a number of cell layers. So examining the number of the cell layers from within outward of the diagram, these again are the cell layers that the oocytes must come across to ovulate. So we'll start with the granulosa cells in this part of the follicle. After that, we have the basement membrane, the one that separates the granulosa cells, and the third layer, which is the theca interna. We also have the theca externa, the tunica albuginea, and the germinal epithelium. Also shown in the diagram are the various hormones that are important for ovulation to occur. So we have here the GnRH, FSH, LH, estrogen, as well as the prostaglandin. The process of ovulation involves various factors. So one of the factor that will determine ovulation is the hormones. Examples of the hormones that are very important for ovulation are prostaglandins as well as the progesterone. So when we are going to block these events in the follicle, it will also prevent ovulation. Number one is the protein synthesis. So there are various proteins that are produced by the follicle that is important for ovulation. Number two is the steroid synthesis. So of course we have the progesterone. Progesterone is important for ovulation. We also have the prostaglandin synthesis. So the PGF2 alpha, for example, is important. It is also one of the factors for ovulation. Remember that PGF2 alpha is also important for CL regression. We also have the synthesis of the plasminogen activator. This is also very important for ovulation. So when we are going to inhibit these events, it will also inhibit. These will also inhibit ovulation. The slides to follow will enumerate the important events that will lead to ovulation. Of course, the very important factor that will lead to ovulation is the preovulatory LH surge. So one of the effect of the preovulatory surge is cumulus expansion. The cumulus expansion will separate the oocyte from the follicular wall that will contribute later to ovulation. So these are the diagram, the close-up look of the cumulus expansion that is brought about by the preovulatory LH surge. So this picture on the left part is the unexpanded portion or the picture of the cumulus expansion. So shown in the diagram are the oocytes that have been aspirated from the antral follicles. The oocytes, as shown in this picture, are located in the center, and they are surrounded by, of course, zona pellucida and the cumulus cells, or the cumulus forus cells. We can also see in the follicle cells, again, the cumulus cells. And again, under the influence of the LH, we get the expansion of the cumulus cells as shown on the right. So this is an expanded part of the cumulus cells. These cumulus cells will produce, or of course, these are the cumulus cells, and the one at the center is the oocyte. These cumulus cells will produce hyaluronic acid and some proteins, and they push apart from one another. This structure grows tremendously with the oocyte remaining at the same size. The next phase include the oocyte and the surrounding cumulus cells breaking free from the rest of the granulosa cells. It then floats free in the follicular fluid of the antral follicle. The next factor that is important for ovulation is the changes in the protein synthesis in and around the follicle. So these factors, now under this, we have the production of the progesterone or P4. We also have the production of the plasminogen activator and the synthesis of the prostaglandins. These include the PGE as well as the PGF2 alpha. The pre-ovulatory LH surge will also lead to the increase in the release of histamine as well as PGE2, which is a form of prostaglandin. The histamine will increase the blood flow to the ovary and the follicle, and it will ultimately lead to edema. Edema will, because of edema, the follicle will become bigger because it will take up fluid in the follicle. The histamine in PGE2 will also lead to increase in the vascular permeability, and this will allow the production of plasminogen, which is the inactive form of plasmin. The plasminogen will be converted to its active form, plasmin, by the presence of the plasminogen activator. Plasmin is of course a protease now that is important in ovulation. We also have the P4 or the progesterone. The P4 now from the follicles will cause the production of collagen. The collagen will be converted to its active form in the presence of plasmin. The collagenase now is important because it is now a factor that will weaken the follicular wall that will ultimately lead to ovulation. So as a summary, all of these factors will lead to ovulation. So we have the expansion of the cumulus. We also have the changes in the protein synthesis. So for example, we have the plasminogen activator, as well as the prostaglandin synthesis PGE and PGF2 alpha. And of course, we also have progesterone. Of course, progesterone is a steroid hormone and will lead to the production of the proteins, now collagenase, that will be important for follicular wall weakness. We also have external factors such as histamine as well as the PGE2 that is, of course, brought about by pre-ovulatory LH surge, and this will cause increase in the vascular permeability as well as edema, increase in the follicular fluid that will ultimately lead to ovulation. To add, the PGF2 alpha will also be very important now for smooth muscle contraction around the follicle. So all of these components would be important now for ovulation. Domestic animals can also be classified according to the manner by which they ovulate. Humans and horses, for example, are spontaneous ovulators because they do not need external stimuli in order for them to ovulate. We also have the induced ovulators. Examples are the camel and the otter. Induced ovulation is when a female animal ovulates due to an externally derived stimulus during or just prior to mating, rather than ovulating cyclically or spontaneously. Example of the external stimuli is copulation. In induced ovulators, copulation will be a major factor for ovulation. The penis in the vagina will, of course, stimulate the nerve endings, and the signals will be then received by the sensory neurons. Stimulus will then reach the spinal cord, and the spinal cord will send the signal to the hypothalamus. The signal will then impact the GnRH release from the hypothalamus. GnRH will be sent to the anterior pituitary. The AP will produce the LH, and the LH will be important for ovulation, particularly the pre-ovulatory LH surge. An example of an induced ovulator is the cat. So in cats, Tom has spines in the penis, and biting of the neck of the female is an important copulatory behavior. So the success of ovulation in cats will be dependent on the number of copulation. Cats with a single copulation will only have 50% chance to ovulate, whereas when there is a multiple copulation, it will have a higher LH surge and higher chance of ovulation. So therefore, in cats, ovulation is strongly dependent on the number of matings in a short period of time. It will also be dependent on the timing of mating. Now, for example, Wild et al. Showed that only 1 out of 12 cats ovulated after a single mating on day 1 of estrus, whereas 4 out of 12 ovulated after a single mating on day 4 of estrus. So it is dependent on the day of estrus or on what day after the onset of the estrus has cat been inseminated or mated. These are the major events in the oogenesis, or the formation of the female gamete. Of course, it all starts with the primordial germ cells moving into the germinal ridge, and they get into the gonad and begin to divide mitotically. They then become the oogonia, then become surrounded by follicle cells. Once surrounded with follicle cells, they enter meiosis and become primary oocyte. Most of the life of the oocyte will occur at this stage in the primary oocyte, and most of them will die and undergo atresia, and they do not undergo further development. So this is also known as the meiotic arrest. So there are two important phases of the oogenesis. The first phase is the growth phase, which is considered to be gonadotropin independent. So the growth of the oocyte, particularly the primary oocyte, will include the formation of the zona pellucida, production of mRNA, synthesis of protein, and formation of the cortical granule. And these are important for eventual embryo development. The second phase is gonadotropin dependent, and this includes nuclear maturation. The nuclear maturation refers to the ability to continue with meiosis, and we also have the cytoplasmic maturation, which is important to accumulate mRNA and protein, and these are critical to the early embryo development. So the primary oocyte will remain as primary oocyte until LH surge. Under the microscope, these are the oogonia that divide mitotically in the fetal pig ovary. As the oogonia become surrounded by follicle cells, they change into primary oocytes, and they begin to enter meiosis but become arrested at this point. These are the primary oocytes in the primordial follicles. So the primordial follicles are differentiated by a layer of follicle cells that surrounds it. This is a diagram of the primary oocyte in a primary follicle. So the primary follicle is characterized by the presence of cuboidal follicle cells that surrounds the oocyte. This slide shows the fetal oocyte development relative to birth as well as fertilization, and of course the stages in the fetal the oocyte development includes mitosis and meiosis. For the cow, mitosis and meiosis occur well before birth, and that is at the first third of pregnancy. So the pregnancy or gestation period in the cow takes about 280 days, and before birth, the fetal oocyte has already undergone mitosis and meiosis. In the case of the pig, we have some mitosis and meiosis occurring after birth, and the gestation period is about 114 days. And for the mouse, all mitosis are finished before birth, while meiosis may occur until after birth. The tertiary follicle, also known as the antral follicle, is the one that will later undergo ovulation. This slide shows the formation of the zona pellucida. So zona pellucida is of course an important structure that is located in the follicle. As the primary oocyte continues to grow, it begins to secrete a zona pellucida as shown in the diagram. So outside the zona pellucida are the corona radiata cells, and the inner layer of the corona granulosa cells that surround the oocyte will send cytoplasmic processes through the zona pellucida, and they make contact with the surface of the oocyte. This is a closer look of the corona radiata. We have the zona pellucida and the oocyte. So there are gap junctions where the corona radiata cells send signals to the oocyte. So during the oocyte growth and before the LH surge, there are important factors that will lead to the oocyte maturation as well as meiotic arrest. So this diagram shows the corona radiata, the zona pellucida, and of course, we have the cytoplasmic processes here and the gap junctions later. So through the gap junctions, the corona radiata can send signals that lead to the increase in CAMP within the oocyte. So one factor that is important here is the cyclic AMP. And of course, cyclic AMP is increased by the presence of OMI, or the oocyte maturation inhibitor. So the OMI is produced by the corona radiata cells, and OMI is important because it will increase the cyclic AMP. Increase in the cyclic AMP will trigger increase in the protein kinase A, and the protein kinase A will promote the conversion of MPF, or the maturation promoting factor, to its inactive form, the pre-MPF. So the MPF is important because it will promote the metaphase. And when we are going to inhibit MPF, so the metaphase, the promotion of the stage towards the metaphase will also be inhibited. So the OMI is again the oocyte maturation inhibitor, and from the name itself, it is an inhibitory factor that will lead to this is actually the reason why the oocyte remains for so long in the immature decade state of meiosis. So it will inhibit the further development of the oocyte, or the primary oocyte, to its mature form, which is the secondary oocyte. So during the LH surge, the first thing that happens is that the gap junctions are destroyed. This will lead to a decrease in CAMP levels in the oocyte, and a decrease in CAMP would also trigger decrease in the PKA, protein kinase A, and the conversion of the pre-MPF, maturation promoting factor, to its active form. The MPF will trigger the breakdown of germinal vesicles, and it will drive the oocyte into metaphase. So that is the resumption of the meiosis. A primary oocyte is formed as a result of meiosis 1, which occurs around 5 months during the development of a female fetus in humans. This new cell has 4N, or 4 times the number of chromosomes, and preparation to create new cells with the 2N number again. So the LH surge will brought about the resumption of meiosis, and this will form the secondary oocyte. During this time, the first polar body will be extruded or emitted, and the oocyte will be arrested at metaphase 2. During ovulation, in ovulation of most species, the secondary oocyte is ovulated, and the oocyte will be transported to the oviduct, where fertilization will take place, or sperm penetration. So when the sperm gets into the zona pellucida of the follicle and interacts with the membrane of the egg, it will trigger the final resumption of meiosis. The metaphase plate breaks down, extruding the second polar body out, and the haploid portion of the oocyte is combined with the haploid portion of the sperm, forming the pronuclei, or the zygote. For dog and fox, they ovulate a primary oocyte, whereas for other animal species, what is ovulated are the secondary oocyte. So the sperm, of course, will get into the oocyte, will penetrate the oocyte and penetrate the eggs. They do not form a zygote right away. So this is the reason why we have differences now in the timing of insemination, because we have to wait some time for the primary oocyte to become a secondary oocyte. That is the reason why for dogs, for example, the insemination should be done after ovulation, while for most domestic animals, the insemination is done before ovulation.